Thanks to modern medicine, doctors are able to recognize signs in our body to tell them if something is not working as it should. Unfortunately, sometimes it is not that straightforward. By the time these signs appear, damage might have already happened at cellular level, and it can be too late for effective treatments. Complex problems such as cardiovascular diseases, neurological and neuropsychiatric disorders or cancer are areas in which current medicine is limited. We still don't have complete answers to what triggers them, the cellular mechanisms behind their development, and consequently, how to effectively treat them. To fill these gaps, health professionals need to be able to look into how the cells in our organs change, well before the first symptoms appear as the disease evolves, and while applying therapies. But sampling some organs such as the brain or the heart is either impossible or extremely difficult and risky, and it would only give a static snapshot of what's going on. Doctors can't take organs out of the patient's bodies to study them during the development of the disease, but they can do the next best thing: grow small three-dimensional versions of body organs called organoids. To achieve this, researchers use what are known as pluripotent stem cells, made by resetting patients' skin or blood cells into white canvases that can generate any cell type of the body. When triggered under the right lab conditions, they can be grown into organoids that present the key cell types and also some of the tissue architecture of the actual organ. Furthermore, scientists can also grow organoids directly from a piece of tumor tissue to study it at very high resolution. Organoids are thus a powerful tool, and that is why they are at the core of Lifetime, the ambitious European initiative that brings together leading research institutes and multidisciplinary teams to transform healthcare through this cell-focused medical perspective. For example, specialists on organoids from the University of Milan, the Human Technopole, and the European Institute of Oncology in Italy are growing brain organoids to understand the cellular mechanism behind brain disorders. Organoids are already proving key to understanding disorders ranging from genetic developmental conditions such as autism and epilepsy, to the impact of toxins and viruses on brain development. And much effort is aiming to make them just as useful for neurodegenerative diseases like Alzheimer's. This is thanks to organoids being personalized models with the same genetic information as the patient they originate from, so as to provide a platform to observe the different stages of disease at a cellular level and understand the causes behind it, including genetic and environmental influences. A method to replay organ development and the molecular processes that can affect it in each patient, and a tool to discover new treatments or test existing therapies tailored to each patient, as well as testing environmental pollutants to safeguard public health. The Lifetime Initiative provides the framework for organoids to be developed and integrated alongside the other two technologies at the heart of the project. Single-cell technologies will allow researchers to study at once and in the individual cells within organoids how all our genes and other molecules respond to disease, producing huge amounts of unique patients' data. All this information will then be analysed by artificial intelligence to identify disease patterns and individual patients' profiles. Ultimately, researchers expect to be able to intercept developing diseases through cell-based prevention strategies and design precision treatments. This will make Europe a leader in biomedicine and revolutionise healthcare for the benefit of society.